Hangin' at the Reef is brought to you by Hawaii Pacific University's Holomua Commitment, making private university education affordable and accessible for Hawaii students. Learn more about the Holomua Commitment at hpu.edu/holomua.
Aloha and welcome to another episode of Hangin' at the Reef, the podcast where we dive into the stories, voices, and experiences that shape life in Hawaii and beyond. I'm your host, Jeffrey Rich, and today's episode is a special one, spotlighting the intersection of science, community, and the environment, and how research can shape the way we care for our natural resources. Joining me for today's conversation are two inspiring scientists from Hawaii Pacific University. First, Dr. Carmela Vietze, Assistant Professor of Environmental Science. Dr. Vietze is a bioecochemist whose research spans water quality, wetland conservation, and the ecological impacts of bioenergy crops. Also with us is Dr. Olivia Nigro, Associate Professor of Biology. Dr. Nigro is a microbial ecologist whose work explores how microbes and viruses interact with their environments, from Hawaiian ecosystems to extreme habitats beneath the ocean. She's passionate about helping students discover the invisible but powerful role microbes play in shaping our world. Together, they share insights from their research on Genki balls, a popular method intended to improve water quality by introducing effective microorganisms into local ecosystems. Their findings challenge assumptions, showing how these interventions can sometimes worsen water quality and reduce oxygen levels, and they emphasize the importance of scientific evidence, ecological awareness, and community collaboration in conservation efforts. This is a story about looking deeper, asking hard questions, and ensuring that environmental solutions are rooted in rigorous science and community trust. Now here's Dr. Vietze and Dr. Nigro with the conversation. Well, welcome to the show, Olivia and Carmela. We're really excited to have you guys with us today. Your research is making a lot of news locally, and I imagine even internationally as the word gets out about the research that you've conducted on the Genki ball. This is our Genki ball episode. So, let's start with an assumption that our listeners don't know what a Genki ball is. So maybe we could start with talking a bit about, how and where and what are Genki balls and how are they used in Hawaii?
Okay, so I think Genki balls first started being used in Hawaii in around 2019. They are a ball made out of dirt and rice bran and molasses that are meant to act as a vehicle to deploy effective microorganisms into the environment. So effective microorganisms, or EM is the abbreviation for them. The Genki ball is this mixture of components you add to it. So when you see these Genki ball activities happening, people are mixing these components, adding EM, and then normally what happens is the Genki balls are allowed to cure for 2 weeks. So if you go to a Genki ball event, you're actually throwing in a ball that was made by a different group of people 2 weeks ago. And then your Genki balls go to replace those other Genki balls.
Okay, so the kids, for example, that are buying these Genki balls and making them and thinking they're gonna get to throw it in themselves, it's not actually their Genki ball that's being dropped into the water.
Right. So
Okay.
there's a 2-week curing process where I think the purpose of that process is to allow the EM to grow and multiply within the ball. So the idea is, that you have the soil is very dense microbially. So there's a lot of microbes that live in the soil. And so what I think the theoretical framework is that you're adding the EM, and then the EM is meant to eat the molasses and grow in that ball and become a larger proportion of the ball than they initially were over that 2-week period.
So the effective microorganisms in the Genki ball are feeding on the molasses, that's their food, and that enables them to propagate and expand beyond the Genki ball and begin to affect the water quality.
Yeah.
Okay, that seems simple. So the recipe for the Genki ball, and where did the idea, the understanding, or the validation that the composition of the Genki balls would even be effective from the get-go? For example, where did the idea to put molasses in there come from?
So molasses is a simple sugar that can be used by most heterotrophic microorganisms. So many microbes can grow on molasses. So I didn't invent Genki balls, so I don't know how it came about. The whole concept of EM was developed in Japan, and these mud balls, or they're also called bokashi balls, have been marketed in various places all around the world. The scientific literature to support Genki balls, bokashi balls, mud balls is very sparse. Within the soil, as I mentioned, there's a microbiome. Soil is known to be very, very rich microbially. There's one of the richest environments, the most population dense for microbes. When you're adding the molasses, there's no real theoretical framework that would support that you would select for the EM over the soil microbes.
So it sounds like this might have been sort of a marketing idea from the get-go. So if I'm hearing what you're saying correctly, that the natural microbials that are in the sludge, the water, are no different than the ones that are coming from the Genki balls. So why would an introduced microorganism propagate more than what's already there?
That's a similar but different idea. I'm talking about the actual soil that's used to make the Genki ball. So when you make it, there's soil that's brought there, right? And that soil has microbes with it. So when you mix it all up with the EM and the molasses, kind of preempting some of what we found is that the EM theoretically is meant to establish itself and grow more abundant, but it has a hard time competing with the soil microbiome.
Are they both competing, for example, to consume the molasses that's there?
Right. Yeah.
Okay. So
If they're heterotrophic.
okay.
So heterotrophic means that they can't produce their own food like a plant. They can't do autotrophy, which is what plants and a lot of microbes do. So if you're giving them a sugar source, they're most likely, if that sugar source is that there's a lot of molasses there, then the easiest, most energetically beneficial thing for the microbes to do is eat the sugar, right? And so you will tend to favor those microorganisms.
Okay. Well, that makes sense. Carmela, can you give our listeners a sense of how you approached the research, how it was structured in a way that the results would be considered valid and not questioned?
Absolutely. First, Olivia and I wanted to acknowledge our students and collaborators on this project. Former HPU undergrad and current HPU master's student, Emily Andra, she was the one that led the environmental measurements in the tank experiment. And she actually won 2 awards for her research presentations. Lanya Konyu and Maddie Howe were former undergraduate researchers, and they were responsible for measuring the environmental factors in the field experiment. Some of Olivia's master's students, Justin Todd and Anuj Pawar, conducted the microbial community sampling and analyses along with our collaborators at Mike Rape and Casey Kajihara. And we also even had multiple of our HPU classes involved in these collections as well. So it was really a community effort. And then finally, we had the support of Erin Works at DLNR, who patiently allowed us to conduct a 2-year experiment at Hamakua Marsh Wildlife Sanctuary. So I would love to start with our tank experiment first, because that is the sequence of how these experiments occurred. So we had 9 standard 10-gallon aquarium tanks. They were all filled with water and sediment from Hamakua. We had 3 control tanks that just had sediment and water only. And then there were 3 tanks, each with half a Genki ball. And that's closer to about 1 to 3 Genki balls per meter squared, depending on the Genki ball size. And then we had 3 tanks with 5 Genki balls each. If you want to imagine how much 5 Genki balls is, that's actually about 2.2 pounds of material. And that treatment was 10 times higher as the other treatment, or it's an order of magnitude higher than the recommended dose per the Genki Alawai project website. So that high treatment really was designed to address questions about the appropriate concentration for our field experiment. And plus, we were anticipating critiques about us not using enough Genki balls. So we took before measurements after we constructed these tanks, and we monitored the tanks after Genki ball introduction as well. And so the after time intervals consisted of 2 days, 1 week, 2 weeks, 1 month, 2, and 3 months post Genki ball introduction. So we took several measurements over time, right, to really try to look at the effect of the Genki balls. We measured many things in terms of water quality, dissolved oxygen, nutrients, chlorophyll, and we also examined the microbial community composition of the Genki balls, as well as the water and sediment of the tanks over time. And so we found that the water quality in the tanks that received Genki balls was significantly worse than our control tanks without Genki balls. Generally, we saw lower oxygen, more anaerobic activity, which means without oxygen, higher E. coli counts and higher nutrient levels in the tanks with Genki balls. And that was especially at those higher concentrations of Genki balls.
So I want to make sure our listeners understand this. So you're not just saying that the research identified that the Genki balls didn't improve water quality, but that the water quality actually became worse with the introduction of the Genki ball.
Right. Like we had some things that we measured that were only temporarily worse within a couple of weeks or a month, like E. coli. High E. coli levels in the tanks that receive the Genki balls indicate that probably the soil in the Genki balls could be introducing unintended microorganisms, which may also indicate fecal contamination. So that was a bit of a concern. But the other thing that was also interesting was that even after 3 months after the Genki balls were introduced, we still saw water quality effects that persisted. So just to give you an example, at the end of our experiment, the phosphate levels were still 20 times higher in the tanks that received Genki balls than our control tanks without Genki balls. And that phosphate's at high enough levels that it actually caused algal blooms in some of the tanks. So I think the other things that was really important about the tank experiment is that we saw dissolved oxygen levels drop to 0 in the tanks that received Genki balls. And at the end of the experiment, the dissolved oxygen in those high concentration Genki ball tanks was still like 50% of the control tanks without Genki balls. And so
what
I
would
think this
cause
is,
that? What would have caused that, Carmela? What would create oxygen deficiency?
yeah. So if you think about what's in a Genki ball, it's molasses and rice bran. Those are both yummy sources of carbon that bacteria like to consume, and that process depletes oxygen. So, I think that is we're putting carbon into the system that microbes are going to use, right? That wasn't there before. And so I think the concern with the low dissolved oxygen levels is, that could lead to fish kills. That's particularly a concern for native fish like o'opu or a hole hole, for example.
Right, and that's also the low oxygen is what leads to the odors that people frequently talk about in marshes and anaerobic systems. So when you deplete oxygen, microorganisms use other gases that produce that the metabolism of those gases produces things like hydrogen sulfide and those smelly gases. So really, when you get an anaerobic condition, you're creating the kind of those issues that the Genki balls are really intended to fight.
Making the situation worse. Well, Carmela, continue on. You were talking more about the structure of the study and some notes that you wanted to make sure our listeners understood about how you approached the research.
Yeah, so Jeff, I think you were asking a little bit about mechanism for explaining the lower oxygen levels, which we think are likely due to the carbon that are in the Genki ball. But in terms of the phosphate, I think we're not sure whether it's the soil that's used in the Genki ball that's a source of available phosphate, or whether that phosphate is being released from the tank sediment and the Genki ball through anaerobic activity as the oxygen lowers. And so these results actually echo 2 peer-reviewed studies that were conducted by Mikkel Lerling and colleagues. And they essentially examined the effects of bokashi balls, which is another word for Genki balls. And they too found increases in phosphate and decreases in oxygen.
And then additionally, when we look at the microbes, so our study, we were actually able to sequence all of the microorganisms in the water and the sediment, as well as in the Genki balls. And what we found, what I alluded to earlier, was that less than 3% of the microorganisms in the Genki ball were EM. So the EM are meant to go into the Genki ball and become effective, right? Become important members of the community. And what we found is that they're less than 3% in the Genki ball. And then very few of the members of the EM community were present in any of the treatment tanks.
So is there And you mentioned earlier in the conversation that you've already seen that there is not a residual long-term effect where there's a decrease in water quality, for example, but then over time, as the Genki balls do their thing and the microreals and all propagate, that things improve. And you're saying that's not the case either. So over a longer period of time, does the water quality truly continue to decrease, or is even that a short-term impact? And eventually it comes back to its natural state, which still may not be cleaner than when the Genki balls were deployed, but even the negative impacts on water quality sort of resolve themselves and you're sort of back to zero.
Right. Well, so a tank is a unique environment that doesn't have the introduction of new flow or the opportunity for rainfall, et cetera. So I think actually for a more realistic interpretation of what would happen in the environment, our field data would probably be more informative. Carmela, if you want to pivot
Yeah.
to talking about our field data.
You're sure. So we did a 2-year study at Hamakua Marsh, and we had two main locations, Hamakua and the nearby Waiawea. And we had 5 sampling sites at Hamakua and then 3 at Waiawea. And we had 1 deployment site at each of those locations, while the other 6 were at varying distances from Genki ball deployment. And so we conducted 1 year of monthly before measurements in 2023. And then in January 2024, we actually deployed 100 Genki balls in a 100-meter squared area at both the Hamakua and Waiawea deployment sites. And then we deployed an additional 100 Genki balls 2 weeks later at each of those sites. And this frequency and concentration really was based on the recommendations from EM Hawai'i LLC. And we also were using the data from the tanks to know that there could be something as too much Genki balls and that could actually decrease water quality. So we conducted our after measurements like 3 days following the first deployment. And then we took them 1 week, 2 weeks, and then did monthly measurements until the end of 2024. And so we really measured the same water quality and microbial community composition indicators as we did in the field experiment. And as you might expect, right, in a dynamic ecosystem like Hamakua, which is subject to tides, regular opening of the sand plug near Kailua Beach Park, and also urban runoff, there's a whole lot of environmental variation in these factors, but we didn't see any improvements in the water quality after Genki balls were added. And statistically, Genki balls made no difference.
So you're talking about 2 different scenarios, one in a controlled environment with tanks where there isn't replenishment of water, replacement of the composition of what's in that tank. You're measuring the immediate and longer term impacts on water quality when the water is static, meaning it hasn't been changed over. Out in the field, you have the ability to look at how the impacts of tides coming in and out, introduction of natural materials that are going to flow into that water ecosystem eventually anyway are impacting with the Genki balls, and the replenishment of all of the composition of those materials, you're saying sort of neutralizes the Genki ball's impact anyway. So there's no impact from the Genki ball itself. And then as the water replenishes itself and natural things occur within that area, it's really a neutral proposition at that point.
Yeah, that's exactly right. So we see changes, but we see no impact. So we can statistically separate those two things. And although we see changes, there was none derived in the particular sites where we deployed the Genki balls relative to control sites.
Okay. So let me play devil's advocate for a second and ask you, as the researchers, if you were on the other end of the discussion trying to poke holes in the research, are there things that you can think of as researchers that you still need to investigate that could potentially change the outcomes of your research findings? Or is that just not the case because of both the control environments and the field experiments that you've done, that there isn't, for example, a change in one of the controls that could impact the study?
So first, there's some results that are broadly applicable to Genki balls. Then there are some results that are more specific to Hamakua. So in terms of the variability in the water quality and the effects on the water quality at Hamakua, those generally are true for Hamakua. The part that is more broadly applicable is some of the sequencing results that showed that the Genki ball itself does not contain significant or large amounts of the EM. So the Genki ball in itself is meant to be a deployment tool for the EM. So the first critical step is that the EM establish itself in that ball. And if that's not happening, then
They can't propagate beyond the ball.
correct. Then
And-
any study downstream is going to feel the effects of that.
Is there a potential change in the formula of the Genki ball that could conceivably change the outcome of its impact on the water quality?
I don't know.
That would need to be researched, I imagine, and probably take a significant amount of research and development over a period of time to determine what the right formula might be, if there even is one. But it sounds like what your research is determining is, putting that question aside, the current composition of the Genki balls as they stand today are not helpful in cleaning water quality, and that would seem from your research to be somewhat indisputable.
I mean, one of the beautiful things about science is that we can always refine and improve and do things better. And so I don't like to talk in absolutes about science, but I think that a logical conclusion from our data would be that the Genki ball is not an effective deployment tool for EM. I know there has been some discussion about directly deploying EM into the system. But again, all of these different ideas, I think Carmela and I's main message is really all of this should be tested before it's deployed in the environment. So if there were a new formulation of EM, what we would love to see is that was studied, well understood, well characterized, particularly in Hawaii, in Hawaiian waters and Hawaiian sediments, right? And for that to be established, published in a peer-reviewed journal, and then deployed in the environment. That would be, I think, the ideal progression of any kind of bioremediation.
So isn't that usually the case when you've got, government oversight from, a Department of Natural Resources and Land Management, that any deployment of introduced materials into an environment needs to be thoroughly researched and determined to be positive in its impacts? And it sounds like that maybe was never done from the get-go to determine what the impact might be of these Genki balls.
I think that's something to aspire to in Hawaii. I think we had a permit to do our Genki ball deployment from DLNR. I do believe the Genki project has a permit. Last I checked, though, I think it might have expired in 2024. We can't speak to the state regulations or how managers make decisions, but we think it is important to have scientific evidence that supports these methods of restoration. Absolutely.
Wow. I know there was some anecdotal evidence that was stated that talked about visual, improvement in the clarity of the water. Is that there were natural occurrences or things that dredging, for example, I think was cited as a possible reason why there was some improvement in water quality over the period of time that it was being studied?
In the LOI?
In the LOI specifically.
Yeah. I mean, so I think it's very challenging to visually assess water quality, especially somewhere like the LOI, which is so dynamic. I worked there for 2 years, going down there routinely and sampling. And some days it was very clear and some days it was very turbid. And this can vary due to rainfall, the time of year, the amount of nutrients. All of these things can alter it. So in order to do an assessment and attribute it to an external factor like Genki balls, you would need a controlled before data set and a controlled after data set, right? So the anecdotal evidence you could attribute it to dredging, for example. People, I think they dredged in 2020.
I think 2019 and 2020.
Right. Initially, after dredging, you're going to see some improvement in water quality. So it's hard to say where that anecdotal evidence came from. And then similarly with the animals, people seeing animals in the LOI, that again, there's no real before and after data. So this was the frequency of sightings before and the frequency of sightings after.
Okay, so those are all anecdotal evidence, right? And you've now built with proper research methodology, using control groups, an appropriate research study that really tries to quantify the impact of the Genki ball. So your research is met with skepticism by those involved in the Genki ball deployments. What's been, the reaction from the officials that are charged with water quality?
So Carmela and I had the opportunity to present to DOH and DLNR, and they were by and large very supportive and appreciative of our research. Yeah, so I think that our next steps are probably to write a report, either in the framework of a technical report or a peer-reviewed publication that would further allow our research to be evaluated by public officials.
What's next for you in terms of the research? Are you going to continue to try and develop your research in a way that it combats some of the objections that have been raised to either the methodology or the approach that you're taking?
I think I would say kind of echo what Olivia said earlier, right? In any scientific study, there's always room for improvement. There are things you would have done differently if you had more funding or more people power, or even if you knew what the data were going to look like, right? But I think with that in mind, I think we're proud of the rigorous study that we've conducted in, a tank environment and in a field environment, and that, it's significantly more rigorous than what we see on the Genki Alawai project website. And so I think with that in mind, I'd ask that those critiques of our research be equally applied to the data that it is on the Genki Alawai project website. And also, I believe that the burden of evidence should be at least equally high for the company who is making profit by selling the EM.
That's a really good point. And just to make sure our listeners understand, so there is there are ongoing deployments of Genki balls happening in various water bodies, particularly the Alawai, which is one of the big water bodies of water of concern here. So those continue to happen. What are the risks to the environment if this continues to go unchecked and there's no change to the status quo and we continue to deploy these Genki balls?
Yeah, I mean, in my opinion, the worst case scenario would be introduction of a microbe that we didn't intend to be there, right? That could be the worst possible effect, in my opinion.
And so we've seen what has happened around the world when other invasive species have been introduced into ecosystems and become dominant and thrown off the balance in that ecosystem. Here, it sounds like there could be elements in Genki balls introduced that are not natural to that environment. Would that depend on where the Genki ball is being manufactured and, what dirt and mud, for example, are being used to build it?
Absolutely. So each soil has its own unique microbiome, and that will vary depending on where it's from. So we don't know if the dirt came from Home Depot or came from Hawaii, right? Yeah, that's an unknown right now. And so that could be a source of non-native microbes. And then the EM itself, we're working on characterizing the community of the EM itself currently. Yeah, it's unclear kind of the natural history of the EM, if you will.
Is there a risk, for example, if the dirt that's building the Genki ball comes from the North Shore and it's introduced on the South Shore, is that alone a risk? So it's not just outside of Hawaii, if it's coming from the mainland, from a dirt farm in California, for example, that has elements that are not natural to Hawaii, but could also be moving things from one part of the island to the other.
I think currently there is some concern around the state about transferring soils due to some of the fungal and insect outbreaks. There's been this renewed interest in transporting soils like between islands. But yeah, the repetitive, deliberate introduction of non-native material to an ecosystem does have the potential to introduce non-native species.
So that seems like a risk, and as we said, bad things have happened before when that scenario has played out. So Carmela, I know that the folks involved in the manufacturing and deployment of the Genki balls claim that the sludge depth has an impact on the outcomes of the deployments. Can you talk a little bit about your research and how it addressed that question?
Yeah, well, the Genki Alawai project website doesn't clearly define or describe how to measure the sludge, but I take that it's smelly organic matter. And so that's one of the claims that they make, that Genki balls improve water quality by reducing the amount of sludge. And so that's probably the data that the success of the Genki Alawai project is most based on their website. And so when I asked Hawaii representative about how they actually measure it, they told me that they stick a meter stick into the ground. And so to me, that's not a scientifically rigorous way to measure sludge depth because you can't really control for the strength of the user, right? Or the person who's applying pressure to that meter stick.
But would that assume that they are measuring at least in the same physical space each time?
Also that, right?
Right.
And so, yeah, I think when you also think about this in like deeper systems or like in Hamakua, where the water can be like up to your chest and flood your waders, this wasn't really a feasible measurement because you also just can't see to the bottom of some of these systems. And so, I mean, the other thing too is like if you think about sediment a lot, it's kind of ignoring what we think of as like sediment particle size, right? So you might imagine you have this sediment that's mostly clay and doesn't have maybe a ton of organic matter, but you could push a meter stick pretty deep into that clay. That's not necessarily an indicator of really smelly organic matter. And so for us, we actually measured what's called sediment organic matter directly, and we didn't actually see any decreases in our tank or our field experiment. And I think that's a direct rebuttal to their claim that it has decreased the sludge depth so much in the Alloway.
True. And depending upon the waterway, the Alawai for sure, there are periodic dredgings of those water bodies, which reduces the sludge. So any measurement over time would have to account for some quantified measure of how much sludge which was removed in the first place, which I would imagine would be hard to do.
Yeah, and there's also constant sedimentation from Manoa Stream in the Alawai.
Yeah,
So
which is also affected by global warming and environmental impacts, which create more rain or less rain, more runoff, less runoff at periods of time. So all of those are variables that can't be controlled for.
Right. And it really stresses the importance of having a control site or some area that is not treated with Genki balls that has similar environmental processes, right?
Yeah.
In order to compare, that would be an ideal study design.
So I know you're getting ready to publish your findings in peer-reviewed journals, but are you continuing on with the study with adaptations to adjust, or is this really the end of the study pending some new question that you're going to research within the context of your findings?
Right. I mean, I think it's a little bit up in the air right now. Carmela and I both have other projects that we're really, really interested in. And so I think that we would love to do enough research that we have answered some of these questions definitively. So yeah, I think we're both willing to explore doing what it takes to do that. But it is, it takes a lot. It takes manpower. It takes money. It takes energy. It takes intellectual power. So yeah, we have to get all those resources.
What recommendation would you put out there to the public in general, as well as the civic entities that are charged with guarding the quality of our water? Do you have any thoughts or recommendations to convey to them?
The good thing about the Genki Ball project was raising that community awareness and getting other people involved. We would just hope that same spirit of conservation and investing in our water quality would be maintained. And that's, I think, really the driving goal of both of Carmela and I's careers, right? Is to ensure that these resources exist in the future.
Absolutely. And I think the teacher in me wants to encourage all the students out there to, go examine the data on the Genki Alawai project website, think critically about it. How many before time points are there? Is there a control? How, is the distance from the Genki ball deployments clear and the timing of those deployments clear? Is there enough data to make conclusions? You can download the Surfrider data that they actually allude to on their website and compare your own calculations to theirs. So I would just encourage folks to put on their critical thinking caps and just, dive into that data. It's a great classroom activity and it's great, for fostering discussion about, how do we think about data in this age of social media and artificial intelligence, right? And how do we as teachers make sure that we are passing on those critical thinking skills to the next generation?
And I think that's certainly the key to all of this. If you think about what Olivia mentioned in terms of the positive community awareness that this has raised about water quality. And I think about, having younger people, school-age kids, as a class project, build these Genki balls, deploy them as a class activity. And I don't think anybody would argue that that is enormously valuable to seed the idea in young people's minds that you need to be focused on our environment and be doing things that help the environment. And when we can do things that counterbalance negative impacts in our environment or can set things on a path for improvement, that those are all really, really important to do. And that would also make me think that if the impact of a Genki ball was really neutral, it didn't harm, but even if it didn't hurt, you could make the case to say that the awareness building that the project does is valuable in and of itself. But let's be clear for our listeners that what you're saying here is that that is not the case, that the water quality is not only not improving, but becomes degraded, which impacts potentially the environment and its ability to continue to replenish itself and renew itself. That alone is a reason to question the deployment of the Genki balls and say, even with the positive impacts of awareness building, the negative impacts to the environment outweigh that. And there are probably other ways to build interest and awareness in environmental issues than to deploy elements that do harm to our environment.
Yeah, I would agree with that. But I would also add that, we want to make sure that the methods that we invest time and energy in as a community are the best methods. So doing all this work and, our studies, et cetera, we want to make sure that we focus and rally around procedures that are going to make a difference, going to make a change. And, in the world right now, there is somewhat of a mistrust of science. And I think being very ethical in how we report science and how we talk about science is imperative to maintain the trust of the public of scientific studies. So I think that there needs to be a clear delineation between scientific studies and anecdotal evidence and, non-scientific interpretation of data in order to maintain the faith of the public in scientific endeavors.
Yeah, I think that is so critical, especially in the moment in time that we're living in right now, where science is being questioned and facts aren't facts, apparently. So I think I'm very interested to see how the public continues to react to this research, and most importantly, what those government entities that are there to protect us from the introduction of harmful elements into our environment react as well. When is the research study going to be formally published?
We're writing it right now, so hopefully in the next few months we'll submit it. And then that process can take a while. So you submit it and it's out of your hands and, it can take a few months for them to get back to you. But so I would say 6 months is probably our goal to have it actually published.
And do you have any plans to present the research more broadly for assessment of impact?
We've presented our research 3 times at the Kailua Neighborhood Board Water Quality Subcommittee because they're super passionate about improving water quality in the Kailua area, which is where Hamakua is. And we also have presented our research for 2 years now at the Hawai'i Conservation Conference. And as Olivia mentioned, we've had conversations with DLNR and the Department of Health as well. And so, we're happy to have conversations. That's what this is about, right? To share our findings. In the meantime, we think it's an important issue. And I know Olivia and I, when we talked about what was the most important thing that came out of our research, we talked about how Genki balls can have unintended effects that are harmful to the environment or even to public health. And we just really hope that the state and EM Hawaii will collaborate to ensure the efficacy, but also the quality control of these EM products.
Yeah, I think that's really the key at this point. Well, Carmela and Olivia, thank you so much for being our guests today on Hanging at the Reef. And I guarantee you this is not the last conversation we're going to have and not the last that our listeners are going to hear about the big Genki ball controversy that's brewing right now. But I, on behalf of the community and those that care about the environment, want to thank you for your efforts, because I'm sure you didn't go into this intending to shoot down the hopes and dreams of all the young people that, deploy Genki balls and think they're doing something positive for the environment. But, truth is truth, and research shines a light on facts that we all have to take into account when we make decisions. So I look forward to having you back in the very near future to talk some more about the impacts on the water and the reaction to the community and where Hawaii goes next with this really important research and the findings that you've discovered. So thank you.